In the final third part of our conversation about evolution, we're going to talk about the driving forces for the evolution of populations. So we need to remember that neither Darwin nor Wallace knew anything about genetics or genes. Not because they were stupid, but because Mendel's work on inheritance was not publicized enough. In the early 20th century, Mendel's work, along with the Darwin and Wallace views of evolution, were rediscovered and synthesized. So eventually, by 1940s, the concept of natural selection and main concepts of genetics were integrated, producing the very productive crosstalk between the two. Now, you remember in our previous conversation that an allele is a version of a gene that determines a trait. For instance, here there are two tall alleles and the wrinkled and smooth allele, that's for pea plants. So you can calculate the frequency of an allele in a population. Basically, how often a particular allele appears in a population. Evolution is essentially driven by the changes in the frequencies of different alleles. For example, the allele IA for the blood type is present in 26.1% of the human population. When allele frequency changes over time, that indicates evolution. And the driving force that changes the allele frequency is natural selection, because some alleles may be more beneficial than others. Essentially, the changes in allele selection in allele frequencies are based on the traits that increase the fitness of the organism, the ability of the organism to survive and to reproduce. So alleles that are more advantageous are going to increase in frequency, which results in the spread of beneficial traits beneficial for the survival in these particular circumstances throughout the population. I can't stop reminding you all that a trait that is beneficial for a certain environment may be detrimental for a different environment. Populations experience genetic drift, which is just random changes in the frequency of alleles due to the pure chance, not selective pressure. For example, one type of the genetic drift is the founder effect. So here you can see a founder population that consists entirely of, let's call them red organisms, while in the original population there were both green and red organisms. Eventually you see red took over for some reason. So when this small group of the original population starts to new, and you can see all three of them have different frequencies, we just randomly pick populations that contain certain percentage of a certain allele, that results in populations with different allele frequencies. What I want to highlight here, so imagine you have a population of animals that are isolated on an island, and this particular fragment of the parental population has different allele frequencies. Whether or not this population will survive, we don't know. If they have different allele frequencies and these certain alleles become beneficial in this the environment that is on that island, then that will lead to speciation. Now the gene pool of populations is following the Hardy-Weinberg principle of equilibrium, which postulates that if there are no evolutionary forces that act on the populations, there are no mutations, there's no migration, emigration, genetic drift, or any kind of natural selection, then the frequencies of the alleles and genotypes remain stable over generations. So we will have to now to turn ahead to probability concepts. So the frequencies of two alleles at a particular locus in a chromosome are P for one and Q for another. They sum, P plus Q is 1. It's either 1 or another. Now, the genotype frequencies can be calculated following the product and some rules. So the chances of two dominant alleles on homologous chromosomes is p multiplied by p, so p squared. The chances for two recessive alleles on homologous chromosomes are q multiplied by q, so q squared. The chances of the arrangement of two the dominant and recessive allele on the homologous chromosome can be seen like this. So if these are two homologous chromosomes, you can either have this or this. So there are two possible combinations for heterozygotes. So it's p multiplied by q multiplied by 2. And the total frequencies of genotypes must add up to 1. So, for instance, here you can see that the number of individuals, total number of individuals, is 500. So, how many alleles? What is the frequency of Y allele? So we have 245 individuals carrying two dominant alleles, which means there are 490, 245 by 2, dominant alleles in a population, plus 210 from heterozygous, so 700. In terms of recessive alleles, we have 45 individuals, so multiplied by 2, 90 alleles from homozygous recessive, and 210 recessive alleles from heterozygous individuals, so 300. So the total number adds up to 1, because we divide 700 dominant alleles over 1,000 total. Each organism carries 2 alleles, so 500 organisms will carry a total of 1,000 alleles for a particular locus. So we follow that the sum of the frequencies is 1. Now we're going to do the Hardy-Weinberg analysis. If the genotypic frequency for homozygous dominant is 0.49, just like we observed in the population. The genotypic frequency for homozygous recessive is 0.09, just like we observed. And the frequencies for heterozygous, when added together, 0.42. So predicted frequency matches the actual frequency, which tells us that this particular population that we're looking at in this diagram is in Hardy-Weinberg conditions. Not a single actual population meets the conditions for Hardy-Weinberg equilibrium. Instead, Hardy-Weinberg equilibrium is the reference point to detect evolutionary change. When you observe deviations from the expected frequencies of alleles or genotypes, that means that population is under the action of certain evolutionary forces. For example, genetic drift, gene flow, or selection. Hardy-Weinberg principle allows us to identify whether the evolutionary forces act on a population and how. So if there are two or more variations of a particular characteristic, can be called polymorphic, for instance, different skin or hair color. So the variation in phenotypes will be influenced by the genetics and environmental factors, because environmental factors will determine which characteristic, which form, which variation is more beneficial. Now, the term heritability refers to how much of the particular phenotypic variation is because of the genetic difference among the individuals. Some changes may not be heritable. So by and large, body mass, being obese or overweight, to a large extent depends on non-genetic factors. Now, the greater heritability of a particular trait increases the susceptibility of that population to evolutionary forces that act on that specific genetic variation. Now, the variability refers to how diverse the alleles are and how diverse the genotypes are in a particular population. As you can imagine, increased large genetic variability is very important in breeding programs for plants and animals, because that will preserve the phenotypic diversity and reduce the chances for inbreeding. There's a term for negative effects of inbreeding called inbreeding depression. So if you look at the illustration on the right, the inbreeding between two individuals that have the same genotype results in the higher probability of deleterious recessive mutations, like this one, the homozygous recessive, which may lead to the health issues in the offspring, suboptimal physical, mental characteristics, and whatnot. So if you think about this, smaller populations are more likely to experience inbreeding. For instance, the population of the country Iceland is roughly 300,000 people, and actually the government programs actively encourage the citizens, Icelandic citizens, to basically find partners outside of Iceland and bring them back, because to increase the genetic diversity within the populations. Now, when alleles change in population, that means there are some evolutionary processes occur. And the factors that can influence evolution include the environmental variation, genetic drift, gene flow, mutations, and non-random mating. So let's take a look at those. First of all, what is a genetic drift? So as you remember, individuals who have advantageous traits will pass those traits to the offspring more frequently. So a powerful gorilla is more likely to become a father and produce more offspring rather than a gorilla that is smaller, so that increases the frequencies of the genes for the larger size. To the contrary, genetic drift is the random change in allele frequencies in the population. So for instance, these 5 rabbits mate and other 5 don't, because they are in the same population in the proper place at the proper time. So, and then these 2 mate, and that completely excludes, as you can see, the allele for albino from the population. So you can see that the smaller population are more affected by the genetic drift. Think about this. If one individual dies out of a small population, it reduces the gene pool much more significantly than the death of one individual out of the large population. For instance, if we have a population of 10 individuals, death of one individual means losing 10% of the gene pool. In a population of 100, it means losing only 1%. Other causes of genetic drift can be bottleneck and founder effects. So bottleneck effect is when a natural disaster, like a forest fire or an earthquake, kills a large portion of the population at random. So you can see this bottlenecking event, which on accident, these little balls get out of the bottle purely by accident. It drastically reduces the gene pool. So genetic makeup of the individuals that survive become the gene pool for the new entire population. These events may reduce the genetic diversity and probably will not reflect the genetic composition of the original population. Another form that we mentioned already is founder's effect, when a small group of individuals, they serve as founders of the new populations. That's pretty common for islands, or when physical barriers divide the population. So this new group of genetic structure, does not represent the original population, and instead forms a new genetic pool with a different gene variability. For example, the population of Afrikaners in South Africa, they have higher incidence of genetic disorders such as Fanconi anemia and Huntington's disease. Why? Because for some reason, when early Dutch settlers arrived to South Africa, they had higher than average proportion of those genes. Other factors that contribute to genetic variability are types of mating and gene flow. So gene flow is the movement of the alleles in the population or out of the population. Basically, migration of individuals of gametes. When we say about migration of gametes, obviously, it is relevant for plants, not as much for animals carrying pollen. So this gene flow alters the gene structure of the population and will increase the diversity. So basically, this idea of Icelandic government to invite partners from other countries with other different genetic background is essentially kind of a forceful gene flow. Now, for instance, in case of lions, gene flow is provided by males that migrate between the prides. Another type of factor that can increase genetic variability is non-random mating. So non-random mating refers to the mating behaviors that are associated with certain traits or certain proximity. So mating can be assortative and disassortative. Assortative mating is more frequent. In assortative mating, individuals that are phenotypically similar are more likely to mate. Geographical barriers can result in non-random mating when access to distant individuals is limited. Environmental factors can lead to non-random mating because sunlight may change the color of the skin, which will lead to different mating patterns. Some animals, such as alligators, can demonstrate temperature-dependent sex determination. So they will have sex determined by the incubation temperature for the eggs. Another phenomenon that must be taken into consideration is phenotypical variation across geographical gradients. For instance, in colder climates up north, animals are larger. So the phenotype of the species may vary with latitude, that's a latitudinal decline, or they may have different phenotypes at different elevations. This is altitudinal decline. For instance, different mountain plants can bloom at different times at different elevations. So when there's a gene flow between the populations, it will actually maintain the phenotypic differences in a gradual way. But when gene flow is somehow restricted, for instance, through the geographical barriers, that results in quick formation of differences, phenotypical and genotypical, and potentially can lead to speciation, as we mentioned before. Now we're going to talk about adaptive evolution. So the purpose of selection is to select for beneficial alleles that will enhance the adaptation to environment. And it works against deleterious or disadvantageous alleles. So it will lead to, well, that's a typo, it will reduce the frequency of deleterious alleles. Let me correct myself here. All of this leads to the adaptive evolution. So advantageous traits in a population will increase in frequency, and harmful traits will decrease. So this ability to pass the genes to the next generation is referred to as evolutionary or Darwinian fitness. So different individuals within the population can be characterized by relative fitness. How fit, how likely is a particular individual to pass its genes to the next generation of offspring compared to its population counterparts. So because of these different fitnesses and various environmental pressures, selection can be categorized into three categories: stabilizing, directional, and diversifying. In stabilizing selection, the average phenotype is favored and extremes are selected against. For instance, robins usually lay 4 eggs. If there are more than 4, chicks may be malnourished because parents will not be able to provide enough food, and smaller number of eggs may result in no offspring because some eggs will fail to develop. The stabilizing selection actually will lead to decrease in the genetic variability within the population. Directional selection is when environment favors the phenotype at one end of the variation spectrum. The most famous example was peppered moth in England. So original population had fairly light color, but during Industrial Revolution, because of the trees that were darkened by pollution, the individuals with a darker color of the body had an advantage because they could blend in and avoid predation, which resulted in a shift of the genetic makeup of the population and therefore shift towards the more favored phenotype. Finally, diversifying selection. So there are 2 distinct phenotypes that are favored by natural selection, and intermediate phenotypes are less fit. For instance, if you have a population in which large males dominate due to the size and force, and smaller males can reproduce because they can sneak and copulate and pass their genes, extremes will be selected for, while medium-sized males who can neither compete to large or small males selected against. Another example, imagine that you have mice that live at the beach and mice that live in the grass. So light color mice will blend with the sand and will be predominant population at the beach. Dark colored mice will better blend with the grass and will be more common genotype in the meadows. And the mice that are medium colored, kind of jack of all trades and master of none, they will have disadvantage in both environments. And finally, the example of hypothetical rabbit population. The albinos will not be suited to blend to a rocky environment, but the gray and Himalayan will blend better to a rocky environment. Now, frequency-dependent selection. So in this case, selection may either favor common phenotypes, in this case it's called positive frequency-dependent selection, or favor rare phenotypes, negative frequency-dependent selection. So for instance, these lizards on the right, they exhibit a frequency-dependent selection. There are 3 throat colors: orange, blue, and yellow. Orange males are dominant. They fight for females. Blue males form a long-lasting pairs. Long-lasting pairs with females. And yellow males are small. They look like females and they can sneak and copulate. So basically, this is the rock, paper, scissors pattern. Orange has an advantage over having blue. Blue has an advantage over being yellow, and yellow has an advantage over orange, which means that the population of these lizards go through the cycles. So when blue males dominate, the orange males are in the advantage. Yellow males become common. Now blue males start getting selected, and when yellow males increase, blue males are favored, and that continues go on and on and on. So this is an example of a negative frequency dependent selection. So negative frequency dependent selection, as you can see, increases the genetic diversity. Positive frequency dependent selection decreases genetic variability because animals select for similar phenotype. Now finally, sexual selection. So we know that there is a sexual dimorphism in many species of animals. The sexual dimorphism, often the result of the different reproductive successes, because males compete more for their mates. Now, to attract females or to outcompete other males, male organisms can develop certain traits like size or coloration or just body features. So for instance, on the left, you can see a male and a female peacocks. Okay, and you can see that male peacock has this wonderful tail, while female peacock is fairly bland. Body size dimorphism, larger female and smaller male, and coloration dimorphism in ducks when male is way more colored. So females have less reproductive opportunities than male, generally speaking, because they have to care about the offspring. So they favor males with traits that correspond to greater fitness. There are several explanations to this possible explanation. First of them is handicap principle. So peacock tail or bright coloration of the duck, those are risky traits. They make those individuals more prone to predation. But the size signals that a male with that particular tail is fit enough to survive through predation, and it makes that male more attractive to females. Another hypothesis is that it can be good genes. The extravagant traits like bright coloration may be linked to disease resistance and overall health. So the reason why females prefer those males is to ensure that the offspring is basically better quality, higher chances of survival. So that concept, both the good genes and handicap principle, traits like large tail, they seem disadvantages for survival, but they signal to the females that this particular individual has more chances of survival and the genes are better. They signal basically the genetic quality.